All right, so in this video, we're going to look at viruses, okay? Now, viruses are an interesting topic when we talk about domains and kingdoms because they aren't really living things, right? So seems kind of counterintuitive to talk about classifying them. But since we are talking about classifying living organisms, we do need to talk about viruses, all right? So some questions that we want to be able to answer by the end of this. We want to know what a virus is. We want to be able to identify whether or not they are alive. I just told you that answer, so hopefully that's pretty easy to answer here. I'll even give you a hint. No. You want to talk about what they're made of. We want to identify how they reproduce, okay? And we want to talk about how viral infections are prevented. So what is a virus? Well, this is the definition of a virus. It is a non-cellular particle that infects and parasitizes cells. So the fact right here that this says non-cellular tells us it's non-living. So non-living. How do I know that? Well, we know that all living things, right, are made of cells from cell theory. So since it's non-cellular, it must be non-living, okay? It's something that causes an infection, right? So, simple question, is it alive? A simple answer, no, it's not. Now, think way back to the characteristics of living things. Number one says, composed of cells. Well, right there, that cancels it out. It's not living, right? It's got to meet the first 7 of the characteristics of life in order to be considered alive. Now, can it reproduce? Yes, absolutely. Is it based on the genetic code? Yes. Does it grow and develop? Well, we're going to put a question mark here. It doesn't really grow, but it can change over time. It can develop a little bit. Does it obtain and use energy? Again, we're going to kind of put a little question mark there. It depends on your definition of using energy. Can it respond to its environment? Yes, absolutely. Does it maintain homeostasis? We're going to put a little question mark there, kind of iffy on that one. Does it evolve? Yes, absolutely. The flu virus is a great example of a virus that changes each and every year, right? COVID-19, another virus that is seeming to change some. When you see a news article that says like a new strain has developed, well, it's talking about a change, right? So what is a virus made of? Well, a virus structure is pretty simple. It's a nucleic acid, so either DNA or RNA, wrapped in a coat, and we call that coat a capsid. Now, different kinds of viruses obviously are going to have different shapes. The tobacco mosaic virus right here, I believe, was the first virus ever identified. It affects tobacco. It turns the leaves yellow, which obviously if you're a tobacco farmer is a bad thing. Here is the influenza virus right there. This one is called a bacteriophage. Bacteriophages are interesting because they are viruses that infect bacteria. Okay, so this is a virus that infects bacteria. So bacteria can get sick, right? They can get a viral infection. Something that we may talk about later when we talk about genetic engineering. We can sometimes use a bacteriophage to introduce DNA to a bacteria and make it do something that we want it to do. And here's an adenovirus right there, the second picture, right? So that's their structure. They're just DNA wrapped in a protein coat. Now, the protein coat has, especially visible right here on these flu viruses, these little teeny tiny glycoproteins, and sometimes they're called spike proteins. And what they do is when they encounter a cell, they interact with the cell membrane of the host cell, and they get it to kind of open up, and that's what lets the virus in. If they don't have, think of it kind of like a key. If they don't have the right key, they can't get into the cell. That virus can't infect the cell. If they do have the right key, though, these little glycoproteins open up the cell membrane for the virus and allow it to get into the host cell. Now, viral reproduction only occurs in a living host cell. They can't reproduce by themselves. There's 2 ways that this occurs. There are lytic infections, in which the virus takes over the host cell and just right away kind of hijacks the whole cell, takes control of all its processes, and just makes the cell into a little manufacturing plant that just makes thousands and thousands and thousands of copies of virus. And they fill the cell up until it's so full it just splits open, and just, thousands upon thousands of viruses flood out of the dead cell and then go and land on a new cell. There's also a lysogenic infection, and this is often sometimes called a latent infection. It's where the viral DNA inserts into a host cell and it lies dormant for a while. And then when the immune system is suppressed, that viral DNA can kind of spring out and produce a lot of viruses. So two different ways there. We'll look at both of those. Now, I want you to watch this video about the flu. This will give you a nice visual representation for kind of some of those things we've been talking about. All right, so take a minute, watch this video. It's, I think, like 4 minutes long, something like that, and then continue with the lecture. So let's look closer at this lytic infection, all right? So in a lytic infection, we're going to start right here. We're going to say we have a bacteriophage, all right? And that bacteriophage is going to land on a bacteria and inject its DNA into the host, right? So here's our host bacteria, right? That orange rod-shaped bacteria. And then the capsid just kind of falls off, right? It's not really doing anything anymore. The DNA that it injected into the host, though, it's replicating. It's making many, many, many copies of itself. And once it's replicated, it gets the cell to assemble it some new capsids, and then it breaks out. So we have 3, right? So there we go. 4, we have new capsids. So new capsid, right? So it's got all these copies of the viral DNA. Now it's going to give them a vehicle to move, right? A new capsid. And then finally it busts out of the cell. So the host cell dies. New viral particles spread throughout the host organism. So you have entering the cell, make copies of the viruses, and the virus tears out of the cell. You can remember that with EMT. Enter, make copy, tear out. That's the lytic cycle. In lysogenic, think later. Lysogenic adds nucleic acid to the host DNA, then it's going to take a nap, then it's going to enter a lytic cycle so that it can release from the host cell. Now, a lytic infection can be a whole lot worse than a lysogenic infection, because the first step, right, the virus is going to insert its DNA, and the DNA gets inserted into the plasmid of the bacterium in this case. So as the bacteria replicates, right, so this is just the normal cell cycle that we've learned about, right? The parent cell makes 2 daughter cells, and then they make 2 daughter cells each, right? So this would be 3 and 4. And so each time it makes a copy, you can see the viral DNA gets copied as well. And so then when the immune system is suppressed or some other trigger occurs, the virus DNA in all of those daughter cells kind of activates, and you get many, 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 many viruses pouring out all at once. Okay, so let's look at the tobacco mosaic virus a little closer. It was discovered in 1897. It was too small to see at the time, but it was identified as the causative agent behind this yellow mottling of the tobacco leaves, which made them useless for drying and making tobacco. When they finally got a picture of it, they found that it was one of these little capsids. All right, this is a bacteriophage, but it was just a capsid. We can see it back here. Let's go back a little bit just to make sure that you know what it looks like. There it is, right there. That's the tobacco mosaic virus. All right, so 1897, that's the first one. And once you start discovering viruses, right, you have to be able to classify them. So we classify viruses based on the type of host they infect. We group them by shape. And so a bacteriophage is a virus that infects bacteria, and we use them in biotechnology research. A retrovirus has RNA in the core instead of DNA in the core. So that's another way that we would classify these viruses. They use something called reverse transcriptase, You see ACE, right? You should think that's an enzyme, and it makes DNA from RNA. So remember when we talked about how to make proteins, we learned that you start with DNA, and then you make an mRNA, messenger RNA, right? And then this goes out to the ribosome. So a retrovirus can take RNA and turn it into DNA using this reverse transcriptase. Right, that's the name of the enzyme that can do that. And so these can be really difficult to treat. HIV is a major example of one of the retroviruses that human beings struggle with. Now, when we talk about viral diseases, it's important to understand that they do still fall under the germ theory of infectious disease that we've already talked about. And just to refresh your memory, right, the germ theory says infections are caused by viruses, bacteria, or microbes that colonize part of the body and, interfere with the normal function of your cells, which can affect your tissues and then your organ systems and then you as an organism as a whole. Viruses are pretty specific to what host organism they affect. So most, if not all, organisms can suffer from a viral infection. Obviously, COVID-19 is something that's going on right now as I'm making this video. It's the reason that we're in, virtual school, and there are just many, many, many different impacts that this pandemic has had. But COVID or coronaviruses, right? COVID, the CO stands for corona. The coronaviruses are pretty common to mammals. They're coronaviruses that infect dogs. And this specific COVID-19 virus seems to have originated in a bat. So it's not uncommon for there to be viruses that affect certain organisms that then don't affect other organisms. So when there is cross-species movement of a virus, that can be very dangerous to human beings. So things like H1N1, which was the swine flu, which started in pigs, or avian flu, which starts in birds, right? Those can be, obviously very dangerous for human beings. Now, if you want to prevent viruses and viral spread, viral infections, you get an immunization. You can't take antibiotics to stop a virus. When you see the word antibody, antibio, all right, anti means, if I can get my pen to work here, against, and bio means life. So antibiotics are good against bacteria because they're living things, but viruses aren't alive. So you cannot use antibiotics against a virus. You have to use an immunization. And we've talked about immunizations before. We looked at immunizations when we did the unit on human health and the immune system. So if you need a quick refresher, go back there and look at that. You can remind yourself about the memory cells, the B cells, right, the killer T cells, and the immune response that a vaccine causes in the body. Now, viruses aren't the only little particles that can cause disease, right? The germ theory tells us, right, if we look back here, that it's viruses, bacteria, and other microbes. It's also prions, which are little pieces of protein that can start to mess with the normal protein function of your body. Mad cow disease is an example. You can look up mad cow disease, or I believe it's Creutzfeldt-Jakobsen's disease, which is if you are a cannibal, if you eat other human beings, it is a disease you may end up getting, which is caused by a prion. There are also viroids, which are just little pieces of RNA that can interfere with your cell function. And similar to prions and viruses, viroids are not considered to be alive. So hopefully you can now answer these questions. Pay close attention to the last two questions here, right? The 2 ways viruses reproduce and how viral infections are prevented. You want to definitely be able to answer those, all right? If you have any questions, reach out and I'll be glad to give you a hand.